Please welcome Vanaheim.
I want to remind everybody to be on time. We have a tight schedule.
Right? Okay. Hello, everybody, and it's a great pleasure to present Vanaheim Bio, the company that works on targeting DNA damage to treat neurological decline. We envision a future without dementia, a world where everybody can spend more time with the people they love. And the way we do that is to target DNA damage. We know that DNA damage accumulates with age and is emerging as a root cause of dementia. Oops, sorry. And what if we simply just remove it and thereby attenuate features associated with the brain aging? At Vanaheim, we develop novel compounds that are able to reverse persistent DNA damage and repair the aging brain. And in fact, we've already discovered several lead compounds. Our strategy includes first targeting rare DNA repair disorder, ataxia-telangiectasia, that displays neurological aging. This disease will act further as a stepping stone towards more broader neurodegenerative diseases. Ataxia-telangiectasia is a standalone indication of being very commercially attractive and with a great market potential. While for neurodegenerative diseases, as we have also heard from the previous talks, the market potential is huge and with over 55 million patients reported globally. So how can we achieve this? We turn to Nietzsche. What does not kill me makes me stronger. This is a statement that is not only true for humans, but also true for cells. It is a response known as a hormetic response. When you treat cells, they are becoming better at surviving subsequent stressors. So we use AI to identify signatures of this response and then screened over 15,000 small molecules to discover top hits. We went through a large number of experiments to test these compounds and then identified top hits that do not by themselves cause damage to the DNA. Our top drug candidate reduces age-associated DNA damage. It reduces DNA damage in cells derived from the premature aging disease patients, including our target disease, ataxia-telangiectasia. This compound also improves the lifespan in fruit flies. And in those flies during aging, the locomotor function is preserved after drug treatment, and in the primary neurons, the DNA damage is reduced. We then further developed novel compounds based on the chemical structure of the original drug, which have high efficacy in reducing persistent DNA damage, selected the lead molecule that shows reduced affinity to the primary target in order to avoid side effects, and then took this lead compound, tested in mice, and showed that it crosses blood-brain barrier and is present in the brain. So the company key highlights includes the discovery of the first molecules that reduce DNA damage, which are patentable, and we have full commercial rights. The mechanism of action is identified, and our early preclinical data, including in vivo and in vitro models, show the promising results from where our lead molecule has been selected. So what to do next? So we're actually actively searching for the pre-seed funding in order to complete our early preclinical stage. And we are very much interested in the development of our product portfolio. And for that, we propose the following major milestones. First, to generate and synthesize novel compounds with higher efficacy and test our already discovered lead molecule in the disease model of ataxia-telangiectasia. Next, to establish the patent strategy and file the IP as soon as possible. And then, in the last, to develop the validated business plan for further seed funding. And to establish our great team, which right now consists of myself, working for many years in the target ID and drug discovery. We also have Morten and Daniela on board. Both of them have a strong understanding how DNA damage leads to brain aging and premature aging disorders. In addition to that, we have a great advisory board consisting of experts in the DNA damage response field, biotech world, and business development. And with that, I would like to thank you for your time and remind you that in Vanaheim, we are making time.
Thank you.
Thank you.
Thank you. Very, very interesting. It will be wonderful to deep dive into the biology, but we don't have time for that. So, but can you maybe just address whether you will be preventing or repairing?
Yeah, that's a very good question. So, like we have in our data shows that both, if you give the drug early in the stage, it can prevent the development of the DNA damage. And we also did experiments other way around. When the damage is there, the compound is also able to remove it. And ideally, we would like actually to stop the progression of the disease at the end.
So even though prevention sounds wonderful,
yeah,
and we would all love to have that, but clinically that is of course a nightmare. So this should just be aware of.
Yeah.
One more question.
I had one question on in vivo validation, but I'm going to ask the other one, which is you had a fancy slide on what you're planning to do. somewhere there. What's attached real life to it? So what are the actual timelines? Because you now seem to be super early in terms of you're basically a university type of spin out phase, right?
Correct.
We're long way to go to seed. So how does that
what's our
integrate
timeline?
into timeframes?
So right now we use all opportunities we have in Denmark because we want to establish the company in Denmark, and in order to seek the pre-seed funding, and then we plan to do within the next 1 year to complete our preclinical data in mice, including safety data, and then including testing in the disease model to show that our lead compound that we already developed can attenuate already the behavioral phenotypes associated with the disease. And this is the first year plan. And next, to find already seed funding in order to perform activities required already for the CMC, like non-tox and regulatory, already further down in line for the IND submission.
Does that answer your question? Thank you very much.
Thank you.
It does.